My topic for the modeling project was model the propagation of an action potential done in an axon of a neuron. An axon potential is a short-lasting spike-like change in the membrane potential that is passed along an axon as a result of a trigger stimulus. Such trigger can be the result of a small change in charge distribution across a membrane caused by mechanically gated ion channels which are responding to mechanical stress or by the action of neurotransmitters on ligand-gated ion channels, in turn making them open. As seen in the picture, there's a dendrite, the cell body, the nucleus, the myelin sheath, and the axon terminal. The neuron is also made up of an axon, which serves as a point that allows for electrical impulses to go away from the nerve cell body or soma. When axons and dendrites are bundled with axons and dendrites from other neurons, they form nerves. Clusters of neuron cell bodies are called ganglia or neuroglia. Next are Schwann cells that can be myelinated or non-myelinated. Then we have the nodes of Ranvier, which are the only sites where the membrane can depolarize to the threshold and generate an action potential. Thus, conduction along myelinated axons appears a jump from node to node called salutatoric conduction. Salutatoric conduction is more efficient, and action potentials only need to be generated from one node to the next, resulting in a much more rapid conduction when compared to continuous conduction. The nervous system also comes into play. The nervous system cells send signals either as electrochemical waves through traveling along thin fibers called axons, or as chemicals released into other cells, which is split into the peripheral nervous system and the central nervous system. The peripheral nervous system includes nerves, ganglia, and Schwann cells, and the central nervous system includes tracts, nuclei, and oligodendrocytes. These sites, or the main function, is to provide support and insulation to axons in the central nervous system, and they can extend 50 axons. When we take a closer look, the central nervous system is interconnected in complex arrangements and transmits electrochemical signals from one to another. When we look at the peripheral nervous system, it consists of sensory neurons, and clusters of neurons are called ganglia, and nerves connect them to each other. Now we take a closer look at the actual action potential itself. We start at negative 70 millivolts, where the stimulus first occurs, and goes up to negative 55 millivolts, where the gate is threshold. This is the initiation or resting stage. Some potassium channels are open, and the potassium diffusion is occurring, which is initiated by stimulus above negative 70 millivolts, or the threshold. The trigger can be as light as a pinprick or sound. The electrical signal rises from changes in permeability of the neuron's axon membranes to specific ions, which are sodium and potassium. Then we move to rising phase, which is where depolarization takes place, referring to the charge within a neuron becoming positive and reaching threshold, resulting in an action potential being fired. This is also where potassium channel gates are now closed. The stimulus causes gates in the sodium channel to open. Now there's a high concentration of sodium outside, and the sodium diffuses into the neuron. Here is also the moment where the electrical potential changes to above 40 millivolts. Now we make it to the repolarization or the falling phase, which is when the neurons return to its resting/relaxed state after it has depolarized from the action potential. The potassium channel gate immediately opens and the sodium channels close. The potassium diffuses out of the neuron, and now the reestablishment of initial electrical potential is at negative 70 millivolts. Now the sodium and potassium pumps are active. After repolarization, we enter hyperpolarization, which dips below negative 70 millivolts. Hyperpolarization is a charge within a neuron becoming more negative and action potential being blocked from firing. Finally, we see it go back up and make it to the recovery phase, where the sodium and potassium channels cannot be opened by stimulus. The sodium and potassium pump actively pumps sodium out of and potassium into the neuron using ATP. Here we can see reestablishment of ion distribution of resting neuron occurring. This action potential acts as a stimulus to neighboring proteins and initiates in another part of the neuron. Waves of action potentials travel from the dendrites to axon terminals. Out of an axon terminal, an electric impulse is converted to a chemical signal or neurotransmitter. And this concludes the end of my project on the model of the propagation of an action potential down an axon of a neuron.